Hello to everyone. Welcome to our course on fluorescence spectroscopy, principles and applications. There's going to be a lot of detailed information in this course because it's intended for use by scientists who are seriously interested in using fluorescence in their research. So we have divided the course up into short segments so you do not have to watch to all of the sections sequentially. Go to our website and pick out the section that you want, and you can review just that. As just a very small amount of history, we've been teaching this course for about 25 years in Baltimore and in Berlin in collaboration with PicoQuant. I've only included the sections from my part of the course in these lectures. So let's get started quickly. What is fluorescence? If any of you are old enough to remember when the space capsules were, you're probably too young to remember when the space capsules would drop in the ocean and they weren't so sure where they would land in those days. They would need some type of signal, and a typical signal would be a bag of fluorescein, which is a fluorescent compound, drop it in the ocean, and you see this bright green fluorescence. There's a lesson to be learned even from this, which is that the bright green fluorescence appears on top of a dark background because light is coming back immediately from this top layer of fluorescein, but the rest of the light is going deeper into the sea, and you don't see it. There are many fluorophores that you encounter in everyday life, and they have a couple things in common. Almost all fluorophores are aromatic molecules. They typically have polar groups on them, which increases their solubility or provides a site for linkage to other biomolecules. So you can pick the fluorophore of your choice. One of the most well-known ones is quinine. And quinine, they say, was discovered by Sir Frederick William Herschel, the Royal Society of London. He noticed a blue glow coming from an extract from a type of tree, and he had no idea where the blue glow was coming from. He simply noted it occurred. Of course, the Brits said this is the first time that fluorescence was observed, but I'm sure if you went back far enough into the Greek literature, you could find examples where the Greeks have reported fluorescence. A short time after that, another Brit named George Stokes, pointed out something very interesting about fluorescence, and that is the color of the light coming from the fluorescent substance was not same as the color of the incident light. He figured this out by looking at the next advance in fluorescence was by George Stokes, or Sir George Stokes, another Brit, and he noticed that the light coming from the fluorescent sample was not the same color as the light that was impinging on the sample. This might be a little bit of urban legend, but the way it's said that the experiment was done was that he used deep blue light through a deep blue piece of church glass as a filter, and then he looked at the bottle from the side. I don't think they had plastic bottles in those days, so he used a glass of wine, and he noted that he saw a blue color rather than the color that was impinging on the sample. And this is referred to as the Stokes shift. And in all of fluorescence, the emitted wavelengths are always longer than the absorbed wavelengths. Jablonski now from Poland, which I'm very proud of, was the next pioneer in fluorescence, and he created what's called the Jablonski diagram. And the Jablonski diagram contains information on everything that happens to fluorophore when it goes into the excited state. Basically, when a fluorophore absorbs light, it goes to a first singlet or later excited state. There's always some type of relaxation down to the bottom of S1 or something to do with the solvent polarity. Then this excited state fluorophore exists for a period of time. And because it exists for a period of time, that gives it the opportunity for things to happen between absorption and emission. So it can emit with a radiative decay rate. It can return to the ground state, which since we can't see it, we don't know much about it. So we just call it the sum of the crossings. There can be quenching, and there's another phenomena called fluorescence resonance energy transfer, where one molecule transfers its energy to another molecule. We'll see many forms of the Jablonski diagram because they are normally made to describe a particular process. Thing about Jablonski was he was a physicist, and he thought about fluorophores as particles rather than as the complex atomic molecules that they are. And this was changed. What type of molecules are fluorescent that we have? Naturally, we can have intrinsic fluorophores. All proteins, just about all proteins, contain tryptophanes, usually multiple tryptophanes. They are fluorescent. They're fluorescent in the deep UV where you can't really see them very much. If a molecule is not fluorescent, like a cell membrane, you can make it fluorescent by embedded a dye into the lipid bilayer. Or nucleic acids are notoriously non-fluorescent, but there are many dyes that bind to nucleic acids, and we call these intrinsic and extrinsic fluorophores. And for natural fluorophores, probably if you go to any bar late at night and they have the lights flashing and people's shirts are glowing in the dark, that's probably an example of natural fluorescence. The intrinsic biochemical fluorophores are the 3 amino acids that are aromatic, tryptophan, tyrosine, and phenylalanine, and a number of enzyme cofactors. NADH is one and is the most useful of the bunch. It's used extensively, as we'll see. Flavins are not used very much because they're usually very weakly fluorescent when bound to proteins. And the pyridoxals, it always surprises me how little they're used, and I think it's because they're so complicated. But the real advance in fluorescence as regards to biophysics, which is what we're talking about, came from Gregorio Weber. He was my mentor, and we can see his picture on the side here. And what Gregorio, or as we always call them, the professor, recognized that if a molecule wasn't fluorescent, you could make it fluorescent by adding a fluorescent group that reacted with the protein in some way, typically with amino groups on the surfaces. So instead of just having to deal with the deep UV fluorescence that comes from proteins, you could put a fluorescein type molecule on it, a rhodamine type molecule on it, or any variety of molecules, and these all have reactive side chains. So this really was the beginning of using fluorescence as a tool in biophysics to study protein structure and function. Now life is much easier. It used to be that you had to make the fluorophores that you needed to label proteins. And here are just a few examples of those. And we can put them in different classes. There's the rhodamines and the fluoresceins. We have a Bodibi class and Alexa dyes. These were all made for a specific purpose, which I'll explain to you later. But the good news is you open up one of many company catalogs and you can find these probes for sale in forms that are reactive, contain reactive groups, and will attach to biomolecules. If you don't want to label a fluorophore covalently, there are other choices. In the case of membranes, it's pretty simple. You simply attach your fluorophore to something that looks like one of the components of the membrane that you're looking at. And these will typically partition into the membrane and provide a reporter group. You'll typically have one of these per thousands of other lipid molecules, so you're not really perturbing the membrane. But you now have a signal from the membrane which would otherwise be invisible under any form of excitation. And a similar thing with the DNA probes. A number of probes bind to DNA. Some of them intercalate into the DNA. Some of them bind to this small groove. But the basic principle of all these dyes is that when they bind into the DNA, they become more fluorescent, which makes them, of course, visible above the background. And in some cases, they cannot be washed out. So you can wash the DNA forever and the probe stays there. So what has happened? I guess I was lucky because I got started in fluorescence at a time where I would call it a backwater technology. It's sort of like before the agriculture was discovered. Everything, all the experiments were done with primitive instruments and difficult to do. But since then, fluorescence and biophysics have simply exploded, and there are now multiple uses for fluorescence. For those of you who watch Star Wars, you might remember, I can't remember what that thing is called. You put it on your skin and it tells you what's wrong with you. But these things that are similar to this exist today for fluorescence sensing. For genetics, you can pick one sequence out of many thousands of different sequences, specifically label them, and see that above the background. A remarkable development has been the development of DNA arrays, where it's possible to put all of the 32,000 human genes on a single slide, spot them on the slide, and by other multiple spotting methods, detect something about each of those genes. And then, of course, we'll talk about single molecule detection, because this tells us a great deal that we could never see from a molecule by looking at its ensemble emission, meaning the emission from thousands or millions of molecules. All of this is possible because we have strong signals that are existing. A couple more examples. I forgot these. Fluorescence immunoassays. If you have a tissue sample and you wish to identify a particular type of cell, you can add to it labeled antibodies. So the antibodies contain fluorophores. These fluorophores don't exist anywhere else in the tissue. After they bind to the tissue, you can see bright spots where a particular cell that has a surface antigen on it is located. And we'll see that there are thousands of different what are called fluorescence polarization immunoassays, which are used to identify drugs of abuse and medical drugs and measure their concentrations in blood. This is one of the most remarkable things that has happened because of fluorescence. You know, way back in about the year 2000, the NIH decided that it wanted to find a way to measure the entire human genome. If you stretch the entire human genome out, and I've forgotten the exact number of base pairs, but it's about 1 yard long, and it would be impossible to identify all of these bases. They're all similar structures, but getting their sequences is difficult. But this has now been made possible with actually for under $1,000, you can get your whole genome done. And it could have never been accomplished without fluorescence, because none of the other methods that were tried besides fluorescence were capable of doing this. So what can we do with fluorescence? I'd like to use the HIV virus just as an example. You might be interested in many things about the HIV virus. You might be interested in binding kinetics, how fast different proteins bind to it. You might be interested in the distances between different proteins in the virus. There could be conformational changes in the proteins when they bind. So the list goes on and on. And the point that I wish to make here is that these processes would probably never be visible by absorption spectroscopic methods. And I have this summarized on this last slide. Let's look at these two panels first. One is the sky in the daytime, and the other is the sky at night. In the daytime, the stars are still there, but you can't see them because they're wiped out by the brightness of the background. So you can only see the stars when the background is dark. And this is why fluorescence is useful. You see it against a dark background. There's another very fundamental reason which makes fluorescence useful, and that is that it's different information content than all the other optical spectroscopic measurements. If you're looking at absorption, it occurs in a very short fraction of a second. All you see is the ground state absorption spectra. If you look at Raman spectra, you can see some molecular vibrations, but it's very weak, and the molecular vibrations typically do not tell you a great deal about what the molecule is doing. But if you look at fluorescence, it's because of this time scale of fluorescence that drops from 10 to the minus 16th or 17th seconds down to nanoseconds, 10 to the minus 9 seconds. This now provides time for the biomolecules to do things. And the things that they can do, they can diffuse translationally, they can undergo rotational diffusion, conformational changes, fluctuations in structure, changes in distances, and all of these phenomena are visible by fluorescence because they frequently occur on the time scale of fluorescence, which is the nanosecond time scale.